Okay, I think it's 10 after, so if you don't mind, I'm going to try to get started. I wanted to, before I get into what we're going to cover here today, I wanted to introduce the two TAs that we have in this course. They will be evaluating you on your presentations, as well as giving you some feedback on your midterm and final exam. As I've indicated to you before, we want this course to be as fair as possible, and I've told the TAs several times to mark fairly, not to be nitpicking too much unless it's called for, and to make sure that you get the best mark possible. So I'll ask the TAs, Bethany and Victoria, to introduce themselves, tell you a little bit about where they're doing their research, and yeah, we'll get started with the lecture soon after.
All right, so I'm not mic'd.
Do you want the mic?
That's okay. I'll be a little bit quiet. I lost my voice. So I'm Bethany. I'm a second-year master's student at Atlanta, which is a public health school here at U of T. I do my research in occupational disease, so I'm interested in prevention of occupational diseases. I don't have my background in genetics or neurobiology. I did study a little bit of that in my undergrad here at U of T. I did an HMB undergrad, and I have TA'd a couple HMB courses in the past, so some faces look a little bit familiar from HMB 302 and HMB 202, so I'm looking forward to this course. It's my first year today, and if you have any questions for me or about the public health school, anything like that, feel free to yeah.
Hi everyone, I'm Victoria. I'm also a second year master's student. I work at ChemH, and my work is a little bit in neurogenomics. So I do psychiatric pharmacogenetics, which is looking at psychiatric diseases and how genetics can affect people's response to psychiatric medication. So this is my first course that I'm taking, but if you have any questions, hopefully I can try to answer them, or if you have any questions about grad school or this research in our graduate program, I can also tackle that.
All right. Thank you, TAs. Thank you. And you'll be seeing them a lot, particularly around the time of your midterm, as well as during your presentations. I do want you to communicate with them. I want you to interact with them, and to really get that kind of feedback. By the way, they just reminded me today, later on in the afternoon, if you're really interested in hearing about research, there is a graduate mentorship program going on. So if you're not sure, if you've never done a fourth-year research project, can you still get into grad school? If you have done your neuroscience undergrad, are you still eligible to apply for molecular genetics or into pharmacology? Those types of questions, and about TAing, how to secure a TA position, all those things are things that you can ask at 4 o'clock today over in the Medical Sciences building in the Physiology Seminar Room in MSB 3231. So I'm going to tell you a little bit today about neurogenomics, sort of the next steps. We're going to spend over the next 2 weeks some time for, getting you ready for the midterm on these individuals here. Some of you who are in neuroscience will know Carl Dijsselhoff. Some of you will remember Ed Boyden, who are the pioneers in a field that is really important, that bridges neuroscience and genetics, called optogenetics. And the one individual that you see up here, Fang Zhang, has sort of worked in their labs, both labs, helped to pioneer optogenetics, and is now working on CRISPR-Cas9. For those of you who are on social media, you'll know that right now Fang Zhang is sort of looked at as the de facto leader in CRISPR-Cas9 mediated genome editing. But right now, he's actually embroiled in this massive fight. Like, scientists generally get along pretty well with each other. Yes? Not so sure. Right now, he's in a really bad fight with two individuals, Jennifer Doudna and Dr. Charpentier in France, and they're looking at who is going to own CRISPR-Cas9. Who is going to be able to say, I am the king or queen of genome editing. And I want you to be thinking about this. This technology, as I'm going to point out to you, actually didn't exist up until a few years ago. And I want you to be thinking about what you could do thinking about what do we need in neuroscience right now? How can we use genetic techniques in neuroscience to make the next big advance? And I want you to be in that position. Before I get there, I wanted to go over some of the ways in which you are going to be evaluated. This is still up in the air, by the way. I want to make sure that you're all okay with this. Some of you were kind of skeptical about whether or not you would actually be able to benefit from this. So if you are writing the individual midterm and you got an 80% on the individual midterm that you're writing on the Tuesday after reading week, you will get a 64% adjusted mark. So 80% of 80% is 64. If you then come in on Thursday and you write a very similar, almost identical, a couple of differences, not completely the same, but you'll still be able to do well on it if you study for the midterm, just as you have. And you then get 100% as a group. Everyone in the group gets 100%. Then that mark is 20%. So your mark will overall go up to 84. In other words, you've gained 4%. Now you're asking, what happens if our group does really poorly? What happens if I started out with 100%? Will I go down? No, you won't go down. You'll stay the same. We always take the highest mark in this course. So we'll take the 100 that you got. If your group mark is lower, even if it's a 98, we're not going to drop you down. You'll stay at 100. Maybe other people in the group will benefit from this sort of group activity. What if you decide that I don't want to write the group thing at all. I think I got perfect. And by the way, you won't get your mark back until the group mark is put in or you've written the group test. You'll get 100%. I'm not going to penalize you for not taking part in that group activity. And it's your choice not to take part. If you're ill or you have something else going on and you have a valid excuse for it, that's different. But if you choose not to show up or you forgot to show up or whatever the case is, you're still going to get 100% on your midterm. But remember, there's also a participation mark and you won't get a participation mark on it. And that's a percentage mark. So by doing this, you're really benefiting yourself up to about 6.5%. Do you have any questions about this at all? Do you like the weighting or should we change the weighting? Do you like the 80%, 20%? Do you want 75%, 25%? This is okay for you? Okay, we'll see how it goes. I am going to try to get you ready for the midterm the best that I possibly can. So today, I want you to actually start engaging in that 3x3 matrix that we talked about. We're going to go through genomics and neurobiology. We're going to talk about genome editing because remember, we talked about muscular dystrophy and we talked about the MDX model mouse. I'm going to go back to that paper on Tuesday and go through that paper with you. But before we get there, I need you to understand some key elements of CRISPR-Cas9. You hear a lot about genome editing. What is genome editing? Why do we care about genome editing? Wasn't genome editing there in the 1990s? It certainly was. Why all the big deal right now? So I'm going to give you a really quick overview of the different techniques. I'm going to go through specifically the CRISPR-Cas9 system and why it's so important right now. There are lots of advantages, but there are also lots of disadvantages to using the CRISPR-Cas9 system, and I want you to be aware of those. And throughout the remaining parts of today's lecture, I want you to remember one thing that is really interesting or surprising, one thing that you think is going to be confusing, and the 3 slides that you think might appear on the midterm, and then we'll take it up at the very end. So right now, if we take a look at genome editing, in the past what genome editing was supposed to be, it was a panacea. One day, when we get technologically advanced enough, we're going to go in and cure everyone of different types of diseases. And that was the goal for the Human Genome Project, that if we knew the sequences of the genes, that we could cure anyone if we got to that stage of any type of disorder. And we got to that stage. We got to the stage where we were able to identify the entire human genome, and the goal was then, knowing the whole genome, is that we would be able to go in and prevent disease. We'd be able to take parts of that gene out and maybe make for that disease to disappear. In this gene therapy approach, where we either change it, rectify it, replace it with a better copy of a gene, or delete the gene entirely if it's something that is deleterious or there's an insertion that's in that gene, where we have disabling mutations, we could go in and correct it. That was the goal. That was the overall theme that people wanted to engage in. It didn't turn out to be that easy. Now, again, I'm going to take this up at the end. I'm hoping, though, that all of you realize that you are not responsible at all for this slide. So this should not be in any way like your 3x3. It's to give you an idea. You hear a lot right now, and I told you at the very end of 2015 that the editors of Science decided to make a statement. It was a subtle statement, but it was a bold statement. Have 3 papers back to back at the very end of 2015 where CRISPR-Cas9 was introduced to the world in terms of in vivo adult genome editing back to back in a disease model that people were trying to cure for a number of years, leading into 2016 the next day. But why are we at that stage, and why do we really care about genome editing? Where did this all come from? In fact, this is actually not a new technique. It's not a novel technique. It's been around for a number of years. CRISPRs were actually first described and published in the literature, and each of these dots represents one publication in a peer-reviewed scientific journal. You can see that CRISPRs were originally described in 1987, and nothing happened with those CRISPRs for almost 20 years. It went kind of by the wayside. So is all science sort of wasted? It's not. And that's one of the take-home messages overall in the theme, that science and research in science has many uses. But genome editing, in advance of the Human Genome Project that finished in 2000 and 2001, actually began much earlier. We knew about nucleases. We knew about restriction enzymes. We could cut DNA at specific sequences, and we knew how to do that back in the 1980s. It was really hard, by the way, to do that, because I remember being a graduate student where I had to go in and we had to do some cloning, and that cloning was actually my entire project, sort of as a graduate student. It was that difficult at the time because PCR was relatively new. The restriction enzymes weren't as advanced as they are now, but we still thought of genome editing back in the 1970s and early part of the 1980s. However, these two technologies, these two ideas, genome editing and this CRISPR, which was described as this bizarre immune system that was found in bacteria, came together in a series of papers almost by accident, starting in 2011. And then there was this one seminal paper that came out. This one paper revolutionized science for all time. You are not ever going to go back and say, CRISPR-Cas9, we don't need you. From this moment forward, in 2012, with this one paper, scientists realized the unique and powerful method of CRISPR-Cas9 to do genome editing. And you can see that from that one paper, 1 year later, so scientists read that paper in 2012, decided to get to work in 2012, and started publishing in 2013, 1 year later, on this technique. So you can imagine you've just heard about this brand new place that you want to go and visit. And it's like almost dropping everything and going off and rushing to this brand new location that you've only heard about. And that's very similar to what you see going on here. You can see this huge crush of papers that came out after that one publication. So genome editing is pretty much here to stay. So this gets into how do we do genome editing? And there are really two fundamental techniques. They're more or less the same. All of them actually involve nucleases. So these are all nucleases. Cas9 itself is a double nuclease. And then we have these other restriction enzymes which are acting as nucleases known as FOC1. And you can see that whether we're using zinc finger nucleases or we are using tail nucleases, as you see here in B, all of them involve cutting DNA at specific locations. And that is the overarching theme, that we can go in and cut genomic sequences just like we imagined we could do back in 1979 and in the year 2000 when the genome sequences were completely mapped out. We would be able to go in and knock out, replace, cut genes at will, and have this sort of golden era of genomics. Now, I'm going to go through each of these methods in some more detail. So if you have any questions along the way, please let me know. I'll be happy to go through them with you. Again, this is known as targeted genome editing. It's not just random. We are targeting specific genes, going in there with programmable nucleases that are targeting specific gene sites. And we can use these zinc finger nucleases, which on the previous diagram was under C, or we can use transcription activator-like effector nucleases using that FOC1, which was in B, also known as talins. Some of you might have heard of talins. Or we could use CRISPR-Cas9, which was at the very top, and the newest member of our programmable nuclease targeted genome editing family. So now, actually, that pipe dream that we had in the 1970s, in 1979, when it was just a dream, the reality is we can literally do this at will. Have we done this? Yes, we have, and I've shown you a couple of instances where it's been done in adult mice, and we'll come back to that on Tuesday. In humans, it's been done in the germline cells, but those experiments were stopped. They weren't allowed to continue to full maturity. The cells at the zygotic stage were stopped. They were reprogrammed, and it was shown as a proof in principle that we could do this in germline cells. And we've also used talins. Have any of you seen anything in the news about talins? Using talins in humans here in Toronto? Oh, I forgot. You're not really a newspaper reading group. That's okay. I'll get you to read the news a little bit more often, I'm hoping. In Toronto, actually a form of leukemia was treated using talins. They didn't think CRISPR-Cas9 was specific enough, and I'll get to why they thought it wasn't specific enough, to reprogram cells, the stem cells, and then put them back into a patient to cure them of leukemia. And it was pretty big news. It made news around the world, and it was at the same time at the end of 2015 where we started to hear about all of this. So right now, these gene therapy tools are looking at, can we not just edit out? Can we actually replace a faulty gene? And that is something that is being done now. The 3 papers I'm going to mention to you again on Tuesday, the ones that were in Science, don't do this, but it's completely possible to do this right now. Can we look at this gene therapy, and can we get rid of some of the unwanted side effects? If we're going in at the level of the germline, we want to fix some parts of the DNA, but we also don't want to have unwanted mutations. We don't want it to be sort of a generalized genome editing. We want specific areas to be edited out, and the other parts that we don't want to touch, we don't want off-target effects. And so this newer RNA-guided genome editing tool, which we call CRISPR-Cas9, and this CRISPR, by the way, in case you were wondering, is this clustered, regular, interspersed, short palindromic repeat system, as well as the Cas9 standing for the CRISPR-associated nuclease 9, is a group of programmable nucleases. In other words, they target specific genes in your DNA, and overall, we think it has a number of advantages. Now, at the very top, some of you who have taken other courses with me, you'll remember a little bit about the Cre-Lox system. Back in the 1980s, this is the way in which we started to reprogram elements of DNA. We would have, for example, LOX-P sites that we could genetically engineer around a gene of interest, and then we could express a Cre recombinase to excise or remove that portion of DNA, or to invert that portion of DNA. And we're going to actually come back and use this really old technology, or relatively old technology, to look at single cell sequencing in neurons to our advantage a little bit later in the course. So you don't need to know all of the details right now for this Cre-Lox system. I'll come back and revisit it or reteach it for those of you who need that refresher. But really, we're looking at these 2 systems here, whether it's the TALEN system here in the middle or the Cas9 CRISPR system here. We still, in both systems, have exactly the same thing. We have a stretch of DNA that we want to edit. We have a nuclease. In this case, it is the FOC1 nuclease that we have to genetically engineer. or in this case, we have Cas9, which are two separate nucleases. Cas9 has two subunits with two different nucleases associated with it. And we have some way to target it to specific gene sequences. It has to be programmable, in other words. It can't be random. It has to go to the place where you want it. It's a lot like trying to map your way to a specific location, and you don't want to be going to the wrong location. And you need to be able to program your GPS to get you to exactly the right area on the genome. So you might wonder, why is this really important for the nervous system, right? So if you can do all of this stuff, like, and you could do all of this stuff with TALENs, like, why are people so concerned about this CRISPR-Cas9? Why didn't people jump on all of the other technologies, some of which I'll mention again now, and some that I'll come back to later? Why do people actually have this big issue? One of this is that neurons are very, very unique cells. Not all the information that I'm going to tell you is up here, but you know that neurons are unique cells. Can you name me anything that's not up on this slide that you know is unique about neurons? Makes it very different than any other cell type in your body. Anything? Anything that you recall from any of your other classes or anything that you've read? So neurons are what we call postmitotic cells, right? So aren't most of your cells postmitotic? They are. But these postmitotic cells actually sit in a very unique part of the cell cycle. It's known as G0. They're sort of sitting there outside of the normal cell cycle, postmitotically, and that makes them very, very challenging to do different genetic techniques on. And I'll come back to that a little bit later. Structurally, these neurons are very different looking than normal cells. They're not these flat cells, they're not these round cells. They have these long poles associated with it. They have directionality, in other words, they have polarity associated with them. And in addition to that, they have a network of proteins known as the postsynaptic density. And I have to apologize, I use the short form that we often use in neuroscience. This postsynaptic density makes it very difficult to introduce genes into specific regions of these cells. So we can't really use the traditional targeted genetic approach. We can't, for example, generate embryonic stem cell knockouts and hope that everything is going to work out the same way. The structural integrity of the neuron itself and things like the PSD make this very, very challenging. If you were to go in and do a global germline knockout, as you probably have heard in other biology, second year or first year biology courses, if you knock out a gene, what have you heard happens in that cell? I knock out gene X. What happens? Gene X is gone. And does anything else happen? Yes. So other genes could be affected. Some might compensate. They might be elevated beyond their normal levels. Do you recall from the last lecture, because that is something that you need to remember, if we knocked out dystrophin or we had truncated forms of dystrophin, was there any compensation in our MDX model system at all? What came up? What was increased in expression? Yes. It was utrophin. So we know already that these global germline knockouts, from the time that they're born, just like the MDX mouse, that sometimes it leads to lethality. Sometimes if you knock out a gene, that animal actually doesn't survive beyond the embryonic stage. And sometimes it leads to compensation. Other genes go up as well. As I mentioned, the Cre-Lox system is a great way to conditionally remove genes, and we'll come back to this later, and this involves introducing Cre recombinases. So I've just went through a whole bunch of slides. Before I move on, do you have any questions or things that you want to go into in more detail? No? Okay, so this CRISPR system, as I mentioned, was actually originally found and detected in people that were involved in the food industry. So it was involved first in yogurt and cheese. When your cheese and yogurt becomes bad, or it doesn't taste the way that it should, it's usually because there's a bad batch of bacteria. That bacteria has become infected. And when they started looking at how do we make sure these bacteria that we need, these lactobacilli that are found in different forms of yogurt, the type of yogurt that some of you are eating right now, How do we make sure that they don't become infected? Bacteria can have an adaptive immune response. If they've been infected before, they will actually be immune to being infected by that same bacteriophage because they remember, they've chopped up the DNA of the bacteriophage, and you'll understand why having small segments of the bacteriophage is useful. And they will also have built into the bacteria their own nucleases. So they've got a nuclease, and if they get infected by another virus or something else, they can chew apart the particular virus they were infected by before because they carry the remnants, the immune response that they originally had. And we now use this idea in CRISPR. That was that original paper back in 1987. And we now have these specific nuclease Cas9s to use along with those short segments. Originally, those short segments when we were in bacteria, now with genetic technology, we can create our own guide RNAs to target specific genes. And in theory now, we can go around this issue. So excitatory synapses, where we want to introduce genetic changes, where that postsynaptic density makes it really hard to introduce genetic changes, and we don't want to go to global knockouts or germline knockouts, this CRISPR technology, in theory, allows us to get around both issues. And I'll come back to that again. Again, I don't want to hit you over the head too many times with the same diagram, but it's important that you realize a couple of things. So we need it to be programmable. It cannot be targeting, or it cannot be destroying, deleting, making changes outside of the region that we want to be changing or deleting or modifying. And so we need some sort of homing signal, and this homing signal comes in the form of this specific guide RNA, which we will abbreviate as an SG RNA. So specific guide RNA that you see here. We build this. We read the DNA sequence, we design this, and there are some technical challenges to designing this, and then we add other parts to it. In order for it to be part of this nuclease, it has to have this particular conformation, and we have to engineer all of this. Have any of you actually been involved in cloning or doing recombinant DNA technology using PCR, for example? Have you done this type of experiment in any of your third year or fourth year labs? It's actually not that challenging. As third and fourth year students in molecular biology or with any kind of biology background, it might sound like it would be difficult, but it's actually a really easy process by PCR to construct this. It's not very challenging at all. And that's one of the beauties of this CRISPR system. And then we have these two different nucleases that I talked about. There's one nuclease up here at the top in this subunit, and there's another nuclease down here in this subunit. Altogether, there are two nucleases, in other words, in this Cas9. So it says Cas9 nuclease, but in fact, there are two separate nucleases. So how do we get this gene into a cell? We can use a virus to deliver it, and we'll talk about that. Or we could use a transfection technique. We can tag our guide RNAs with GFP to make sure that it's going into the nucleus and not somewhere else. And so we have a number of techniques that we can use to make sure that CRISPR is actually working well. So again, sorry, I'm just repeating this because this is a big thing right now. We have this capability and this capacity to be able to use this to our therapeutic advantage. We can program because we know the genome sequence. We now know where some of these disease genes lie, and we can then use CRISPR-Cas9 with specific guide RNAs to program and home in on those regions. So I'm going to go through a couple of specific details related to CRISPR-Cas9, then some of the pitfalls of CRISPR-Cas9, and then into some of the advantages, again, of CRISPR-Cas9. So the main element of this is this Cas9 nuclease. One of the big components is this Cas9 nuclease, two different subunits, as you see here. There are two different catalytically active nuclease domains. One is known as the RUB-C that you see up here at the top. The other domain is known as the HNH nuclease that you see down here at the bottom, and they will target and cut wherever the guide RNA is sitting. So wherever the guide RNA ends up binding, the HNH nuclease will cut that sequence, and then in parallel, the RUB-C nuclease as part of the Cas9 will come in and cut this as well. we isolate the Cas9 from bacteria. And this is really, if some of you have been reading the different social media sites, and if you've been hearing about this battle that's going on between Jennifer Doudna and Fang Zhang, it's really centered around this. They are trying to patent this technology. Who owns this Cas9 molecule? Because Fang Zhang says that he does. He owns Cas9 because he put in the patent first. He had one of the first papers in press about it. And that's one of the reasons why he keeps modifying the Cas9. He makes a new generation of Cas9, or he isolates the nuclease from different bacteria. Someone at some point is going to win the battle for who owns Cas9 as this targeted nuclease that homes in on specific regions of DNA. And again, it is also dependent on creating this targeted site. There has to be this sort of structure that you see up here. This guide RNA has elements that involve parts of the CRISPR nuclease system, and we design the other complementary part that targets the specific element of DNA. Now, one of the other things that you're going to see is that how do we organize all this? Is it any piece of DNA that we can actually target? It is not. Right now, we have some limitations, and I'll come back to this slide in a moment, or another slide in a moment. But one of the big issues is, in order for us to target a stretch of DNA, that stretch of DNA has to contain what is known as a PAM sequence on the opposite strand. And this is that opposite strand. Here's our guide RNA up here at the top. The opposite strand has this PAM sequence that we have to have, and this PAM sequence allows this Cas9 molecule, the nuclease, to sit in the appropriate conformation. And this PAM site that you'll see in the literature stands for a protospacer adjacent motif. Your DNA has PAM sequences built into it all over the place in your genome, but it has to be taken into account because this is how we target this subunit that you see up here, the HNH subunit and the RUBC subunit, to specifically that region of DNA. We need that sequence right now, in other words. Okay, so I think I've mentioned both of these. The one thing that I'm going to add here, these two technologies, and this might be something that you might want to write down. I've mentioned these two technologies, which involve tailins, which we have to design. We still design these, by the way, these tail modules, or the zinc finger modules, using a nuclease, targeting specific sites. We still design them. We still need to know the sequence of the DNA, but they're much, much harder to design. They're much harder to deliver. They are not RNAs or nucleic acids. We actually have to form peptides or proteins to be able to target them to the appropriate region. And because of that, there are two issues. How many of you have ever made a peptide before? Anyone? Is it easy? Ingrid, it's not easy. It's also expensive. If I want to design something and I need to design like 10 of them, just in case like 9 of them don't work and 1 of them will, that's very, very expensive, very time consuming. Designing something that you can build by PCR is relatively easy and very, very fast. The other thing that makes this difficult as well, we can target any gene that we want. We don't need PAM sequences for zinc finger nucleases. We don't need PAM sequences to target these talin effective nuclease modules either. However, we can't really target more than one gene at one time. I can chop one gene out, it's done, over with, and that's it. With the CRISPR system, though, it is entirely possible for us to engage what is known as multiplexing. I can insert in multiple guide RNAs, not just one, but right now up to 3 different guide RNAs in the same reaction, and we'll target all 3 locations on your DNA. So I can knock out 3 different genes. With this one system, the 2 systems down here on the bottom, with zinc finger nucleases and talins, I can't. It's 1 gene at a time. And so it becomes much more advantageous to us to multiplex. If I think, for example, that gene A is important, but gene B might compensate, I can get around that problem by targeting gene A and gene B, and maybe even the effect on gene C. So this is a slide that, again, we'll talk a little bit more about at the end of today's lecture, but I want you to be aware of a couple of things. CRISPR, you hear a lot about it. And yes, the number of labs here at the University of Toronto in research labs that are using this it's actually doubled since the end of last year because it's so easy, it's not expensive, don't need specialized equipment. If you run a molecular biology lab, you can do this right now. You can just order in everything and get the job done tomorrow. But there are some disadvantages that I want you to be aware of. These are some limitations right now. It doesn't mean that there are going to be limitations in 5 years, but there are limitations right now in 2016 for us as research biologists. So right now, we can't target everything in DNA. Not every single sequence in DNA is going to be equally targeted. We need those PAM sequences that I mentioned to you earlier. If your stretch of DNA that you are targeting doesn't have a PAM sequence on the complementary strand, your experiment, your guide RNA is not going to work. In addition to that, you also need a guanine at the 5' end of your guide RNA as well. And for talins, the only thing is you have to have a thymine. That's it. You don't need like these PAM sequences. That's the only limitation. And that's fairly easy to get around. So one of the reasons why I told you earlier, they had their choice. These researchers here in Toronto actually had their choice. How are we going to knock out this cancer gene? We could either use talins or we could use CRISPR. CRISPR is cheaper. I just told you that. Talins take a lot more planning. In order for you to generate these little peptide molecules that will generate that nuclease to specific sites, it's much, much more challenging. Why did they go with talins? Because it's been thought that talins are really, really good. They have the best GPS system. In other words, you target it, you program it for this area, it's going to get you to that area with very, very little off targeting. You're not going to sort of wander around and hit, before you get to the Brunswick House, you're not going to hit the Futures Bakery, you're not going to run over and hit the Spadina subway station, you're just going to go right to the site that you want to go. And CRISPRs are thought to be a little bit more lax, more off target, in addition to getting the job done at the gene. So these talins were used because it's been thought that they have less off-target effects relative to CRISPR-Cas9, and that's because of the way we generate the nuclease, the FOC1 nuclease. Talins right now are also really good at targeting mitochondrial DNA. CRISPRs are not, so that's still a limitation. If we want to manipulate mitochondrial DNA, we might have to use talins in addition to using CRISPR. Can you think of a reason why we would even care about mitochondrial DNA? Why bother? Isn't everything encoded at the level of the genome? The nuclear genome? Yes.
The mitochondrial DNA is a separate DNA because it's given only from your mother.
Right, so it's a separate source of DNA. We know that it's involved in psychiatric disorders. We know that it's involved in many other types of behaviors as well. Right now, CRISPR-Cas9 isn't very, very good at targeting this. It's still something that people want to exploit, and they're trying to figure out better protocols to do it, but right now it's one of those drawbacks. When I was in as a postdoc, so not very many years ago, to be quite honest, about 10 years when I was working as a postdoc, one of the things that everyone wanted to do was, you know what, there's this brand new technology called RNAi. Let's do RNAi. Let's knock down a gene. Let's use RNAi to look at this particular behavior. Let's introduce this RNA interference molecule to look at gene expression. What happens if we knock down the expression of this gene using RNAi? Have you guys all heard of RNA interference in your undergrad? Okay, so I'm not going to go into all of the mechanisms behind this. The effects, though, of RNAi are very temporary. The effects on genome editing are forever. It's a lot like your GPA. It just doesn't go away. It stays with you for the rest of your life, right? And so the effects are temporary. They can come back. They can bounce back, and they tend to be fairly non-specific as well. And they're limited to knocking down only transcribed genes. If you have other sequences, like intronic sequences that we are coming to realize as being more and more important, or enhancer regions in DNA, RNAi isn't great at targeting those, okay? So this CRISPR-Cas9 system has sort of now dominated. People still use RNAi. It's still fairly easy to use, but people are now migrating to this CRISPR-Cas9 system. And compared to RNAi, which I mentioned up here at the top, actually this is more specific than RNAi. Why is that? So I told you talins are the best. Right now they're sort of the cream of the crop, expensive. It's kind of like driving a Ferrari, very, very expensive. Here we have the Toyota Camry and the CRISPR-Cas9, but it's still better than driving a Yugo around, right? This sort of RNAi type thing. That's because it tends to have greater slippage because of the RNA structure that gets generated using RNAi. All RNAs have inherent hairpin structures, and because of their hairpin structures that sometimes you can predict, sometimes you can't, make it a little more unpredictable, not targeting the right areas or the transcribed genes. So in terms of CRISPR-Cas9, here are some basic considerations for you. When you are creating this guide RNA that you see up here, this guide RNA has a couple of things that you need to have associated with it. The GC content, the guanine cytosine content that you are creating in this guide RNA has to be somewhere around 50%. It can range from 40 all the way to 80, but it's around 50%. The number of GCs have to be about the same. The length is actually limited right now as well. The guide RNA that we're generating here in blue is somewhere around 20 base pairs, 17 to 24 base pairs. And in blue here, it's represented as the average of 20 base pairs. That's what we are designing. This part you can't design. It has to be consistent in all of the CRISPR-Cas9 systems. That means because of the relatively small size of the complementary sequence that we're generating, there are going to be off-target effects. Now, people are trying to find ways to get around this, but right now, this is one of those issues that are associated with this CRISPR-Cas9 system. So in addition to that, we think that this technology, however we deliver it, whether it's through nanoparticles, whether it's through viruses, whether it's through other viral systems that are longer lasting, it has important ramifications in immunology and, of course, in neuroscience, as well as in cancer research. It's already been applied, by the way, in cancer. And can you think of one disease that involves the immune system that you might want to be eliminating stretches of genomic material, to wipe out genomic material? Yes. HIV. So right now, it's hard to clear HIV, and it's thought that maybe CRISPR-Cas9 might be able to go in and eliminate that virus, the virus that gets incorporated into the genomic DNA out of certain immune cells. So one of the more exciting things is that it's great. It was shown fairly early on in that 2012 paper to work in cell culture, but we want to work with in vivo models. And this Cas9 system, and I'll get into the SP, that is a bacterial isoform. The SP Cas9 has been used in live mice multiple times now. And we just saw 3 papers, or we saw the introduction to the 3 papers in the last class. But we can inject, in theory, we don't have to inject it directly into the brain, in theory. We can introduce this systemically if it's targeting a peripheral organ like the liver or the kidneys. We can inject it directly into your bloodstream, and it will target the right cells and the right sorts of DNA stretches. So right now, people are looking at how to make this system better. How do we make it foolproof? How do we make it easy for an undergrad student to use? And these different types of vector systems are being applied right now in the brain. What we like to use as neurobiologists, we have a number of different viruses at our disposal. I showed you lentivirus, but generally speaking, if neurobiologists have a choice, they like to use the adeno-associated virus, or AAV. And because it's the type or viral vector of choice, because it's a non-replicating virus, Fang Zhang has actually set up AAV vectors that have built into them this Cas9 system. And again, he's trying to dominate the market, and he's trying to make sure that his system is the one that people use. And by doing so, it makes it very easy to clone things in around it. And in fact, the guide RNA expression cassettes, you literally just replace a stretch that he's already created. It's like having a template system available for you, and these cassette systems where you're generating your own specific guide RNA, you can just pop them in. And it becomes very, very easy to clone now and very, very easy to generate. You can do this literally in one day. You don't have to do a lot of the people. So we'll get back into this in some more detail later. Surprisingly, if you take a look at the literature, because I'm always interested in reading different types of papers associated with this technology, if you take a look at the papers, Fang Zhang's group was not the first to use CRISPR-Cas9 in the nervous system. Do any of you recognize any names up here? No names that you recognize? So this individual here, Roger Nickel, is actually sort of the godfather of electrophysiology. And he was the first person to actually use this in slice culture in living tissue to demonstrate that he could use CRISPR. And this was one of those papers that people were looking at going, why did this get published? It got published because, and it got published in a really good journal, Neuron. It got published because he was the first person to use CRISPR in the nervous system. And that's it. That's the only thing that he was able to really demonstrate. There were a couple of other technical things that he showed as well, but it was really the demonstration that we could remove synaptic proteins. And again, this is Fang Zhang's paper that actually demonstrated you can deliver this in a living adult animal, and you can eliminate different proteins, gene expression in the brain in vivo, and this sort of set off a tidal wave of in vivo approaches as well. Now, I want you to think a little bit about what we've covered. And I'm hoping that you were jotting down some notes. I'm not going to ask you about what you thought was most interesting. Everyone's going to have a different opinion. But can someone share what you thought was the most challenging aspect, the thing that you're going to have the most trouble going back and reviewing later? Yes, Arjun. The different components of the Cas9 nucleus. By the way, you are going to have to know that for your midterm. Yes, absolutely. Anything else that you think is going to be challenging? What 3 slides do you think, based on the numbers that we have at the bottom, because you have the same slide deck, what 3 slides do you think you should go back and review for your midterm? Yes. Slide 13. Do you have any others?
I would have to say the comparisons of TALENs to CRISPR.
The comparisons of TALENs and CRISPR. Okay, so let's go back to slide 13. So you're going to have to excuse me for one sec. Let's see if I agree. So knowing the overall structure, how this works, absolutely. You will have to know that. And then you were mentioning some of the, so did anyone pick this slide here? No one picked this slide? So luckily none of you play Powerball because you would have all lost. Anyways, you would have lost. The odds of you winning were like 1 in 900 and something million or something like that. This is a slide that you should make sure that you are aware of and know some of the details of as well. Specifically that there are 2 different nucleases. Any other slides before I get into the rest of the slide deck that you think would be worthwhile going back and reviewing as part of your 3 for this? Yes?
I would also say 17 and 18.
17 and 18. So this is 17. I would say I would agree with you. So just knowing the difference between FOC1, zinc finger nucleases, and CRISPR-Cas9. Do you see that sort of as a multiple choice test question or a short answer question? Yes. You could ask that as a short answer question, but I will ask you as a short answer question, for sure. 100% guaranteed. I will ask you to highlight some of the differences between these because you need to know that leaving this class. And I will also ask you a multiple choice test question based on this as well. Anything else? 18. So yes, knowing the advantages and disadvantages. Absolutely agree. Do you have any questions before we wrap things up for today? I know it's a little dense, so Tuesday's lecture will actually be a little less dense than today. I will be over the weekend posting up specific slides that you should go back and review this weekend for your midterm. It's up to you to keep up to date with things, even if it's just taking a look at them. Please do so over the weekend, or to highlight things that you're completely lost in. If you're lost, what should you be doing? Should come in and see me as soon as you can, because I'd love to help you out with it and make sure that you are on the right track. Okay, so thank you very much. I'll see you all on Tuesday.